Hello everyone, welcome to what will be pre-lecture 19. This should be a really quick one. We're just going to talk about indicators. So what is an indicator? It's something that you have used in lab at this point, likely phenolphthalein. And what happens is the indicators themselves are going to be weak acids or bases that will change color depending on whether they are protonated or deprotonated. And so in the example here, say we have a weak acid as an indicator that is blue when it is protonated, and if it interacts with water or some other base to become deprotonated, it would turn red. And so an indicator's color doesn't change just out of the blue all of a sudden with pH. Generally changes within plus or minus 1 pKa unit. And so we could also think of that as a pH unit. And so that is to say, basically within, say we were transitioning from pH to 7 to 8 in this situation, in that case, we would need, we would be blue at pH 7, red at pH 8, but everywhere in between we would be something intermediate. And so it's kind of my question for you. What color would the solution be if our pH is equal to our pKa of our indicator? And so think about that for a sec, and the answer, assuming you remember your color wheel, should be purple, right? That we should get an intermediate color between blue and red because we will have both species present when our pH is about equal to our pKa. And so if we mix blue and red together, we should get purple, okay? And so again, you've used phenolphthalein in lab. The reason we like phenolphthalein for a lot of titrations, particularly weak acids with strong bases, is that our equivalence point tends to be somewhere around 8 usually. And so you can see that we start to get our transition between 8 and 9 for phenolphthalein. The other nice thing is that it goes from being basically colorless, right, clear, to a bright pink. And so we can really tell when exactly we're hitting that transition point. But as you can see here, there are tons of indicators out there. And depending on the pH you want to see a transition at, you can basically find a indicator for that purpose, okay? All right, that's it for the pre-lecture today. Hope it was informative. If you do have any questions, please let me know. Otherwise, I will see you in class.